So we'll get started on the videotape. One thing I noticed from watching the very start of the videotape from this morning's or this afternoon's class was that when there's chit chat in the class, it's a little hard to hear the videotape. So I want lots of participation, but I will remind you if it starts being a little bit loud in the class to quiet it down so that the videotape is useful. So the first thing I'll do is introduce your TA, my grad student, Tick. So Tick, just introduce yourself quickly and then you can skedaddle.
Quickly, do a PhD. I did my undergrad in chemical bioengineering, so I took this class as well. My research, looking at using drugs and chemicals to reduce scar tissue buildup around plastics or biomaterials, I should say.
So this is Tick's second time taking the course, and of course he took the course. Mostly he grades, but he's been very nice about offering office hours right before midterms for help. I suspect we can arrange that again.
Yep.
Although, my goodness, I can't imagine what actual times are going to work for the entire class, given our discussion on the first class. So maybe a couple of different times or to set up specific times. Same thing actually goes for office hours for me. Obviously, there's no point in me setting one single office hour for all of you because there's no single time that we're not having class that everyone is available. So again, I just want to reinforce that if you need help or there's something that you're not understanding, please contact me, set up an appointment time, and don't worry if you just drop by, there's a slim chance that I'll be there and or available. But if you email me and suggest 3 different times, then there's a pretty high chance that I can make one of those times and we can sit down and have a chat about it. So I'm absolutely available. Please, if you come by and I'm not there, it's not that I don't want to see you or help you. Please don't be shy about emailing me if you do need help. And same thing, I'm sure Tick is also willing to give a hand. So if you're ever feeling a little bit stuck, it's not, I would say it's not a terribly hard course, but there are some concepts that people can get hung up on. And I'd like to help you deal with them sooner rather than later if you're struggling at all with anything. Okay, any questions about office hours? Or so the other thing that I mentioned was the technology issue, so we're gonna try and get it, just the file size is so enormous. Within the next week, hopefully, we'll resolve some way of getting you the videos for all of the classes. And I am gonna just, because I have it, I might as well just video every single class so that it's available for you for our times. But again, I try and keep it as interactive as possible. So today is our last non-math day, hopefully. And we're going to be just going through the background essentials that I expect that most of you know, but might not be top of mind about biology, and that Ryan is kind of giggling about because he's like, oh God, I don't know any of this. So actually, maybe we'll make Ryan come to the blackboard.
Oh no.
Because what we're going to do, and Tick, you can go, thank you very much. So grab a chalk, and in the house. Nice. So what we're going to do is make Ryan, first of all, draw a prokaryotic cell. So you have to tell him what to draw. Draw a circle. Make it bigger. Actually, make it, I would say make it more of a cylinder, oval. Oval. It's fine. You need some space to put some
Draw
stuff.
like a clustered squiggle.
That's
Nice.
good. Okay.
prokaryote, it's just not membrane bound. So it's where all the DNA is stored.
Okay, what else have we got? Ribosomes.
A bunch of little circles.
Yeah. That's good. And ligaments.
Yeah.
So what do ribosomes do? Proteins.
Make proteins?
Yeah. So they help transcribe RNA into proteins, right? Okay. So DNA gets transcribed into RNA on the nucleoid, and then it moves over to the ribosome where it gets translated into proteins. Okay. So what do we call this space in here? And what's this?
Cell wall.
So there's actually a cell wall.
The wall?
Cell wall, yeah. And actually, slightly inside the cell wall is the membrane, the cell membrane, or cytoplasmic membrane. Oh. Okay, so often people draw it more as a rod shape because a lot of bacteria are rod shaped, but they aren't all. So there we have a simple eukaryote. Okay, big,
Isn't that prokaryote?
or prokaryote, sorry, simple prokaryote. Prokaryote.
That's gonna stay forever, you know.
I know, this is why I resisted being on tape for so long. Okay, so now eukaryote, what should he draw?
Draw an animal cell.
A really big cell, a
Bubble
really big
system.
circle.
Another circle.
circle.
This time without a cell wall because it's animal cell.
Right.
It's just a cell membrane. Why?
There you go.
Nice.
That's pretty impressive, Brian.
I remember some things. It's been a while with some things.
So we might as well start actually. Cece just pointed out that there's no cell wall, but there is a cytoplasmic membrane.
Cell membrane?
Cell membrane. Okay. What else we got? So how should he draw the nucleus?
So draw a circle and then do the curly stuff beside the circle.
Pretty good. So that nuclear membrane actually is a bit of a barrier for doing genetic engineering of cells. So if you're trying to do just insert DNA into a cell to do your genetic engineering, if you need it to get in to integrate with the DNA in the nucleus, that nuclear membrane can be a barrier for that. So there are a lot of people who are interested in doing that. Okay, what else should he draw? Okay. Just sort of little squiggles.
What am I drawing?
Mitochondria.
Okay.
So what are
No,
mitochondria?
I just think I'm gonna
Circle with a light bulb.
Yeah, there you go.
draw a zigzag.
Yeah, perfect.
Yeah,
That's
zigzag.
exactly like that.
Powerhouse itself.
So, yeah. So, Cece said it, what is mitochondria responsible for? Energy. Right, energy. And lots of people think mitochondria used to be little bacteria that are symbiotic. What else?
You're gonna make me draw this, eh?
You could probably just like list those.
You can, if you want to just make a list on the side. Oh, very nice.
You know,
Oh. Good enough?
yeah. Which two types though, Ryan?
Ha.
Now
So-
draw it again, eh?
Just say ER. Oh, from ER. get?
Probably some ribosomes, right?
Yeah. Anything else that's important? Yeah, you can put little, we won't write these down, but a lot of cells have vacuoles. There are a number of other organelles that show up. Those are really the major ones. There's one other one. So eukaryotes also encompass plants, right? So what's the organelle that's particular to plant cells? Chloroplast. Right, so obviously our cells don't have chloroplasts. Plant cells do. That's how they transfer light energy into energy to run the cell. Okay, thank you very much, Ryan. That was awesome. Good
I got
art.
to be an artist for the day. Yes.
Good job, Ryan. Very nice, very nice. Okay, so these are the two major cell types that
I'm
you
not
see.
watching that video.
Which are bacteria, prokaryotes or eukaryotes? How many people think put up your hands if you think bacteria are prokaryotes. Put up your hands either if you're not sure or you think they're eukaryotes. Okay, yes. So, all bacteria. And this, we already said animal and plant. What else? So we've got animal, human, plant. Any idea what other cells count as eukaryotes? So yeast and fungi actually also are eukaryotes. And one of the consequences of that is that although yeast are more or less single-celled organisms, they actually are a little bit closer to animal cells in how they produce protein. So some people will argue that yeast are better producers of complex, properly glycosylated proteins than prokaryotes are. So size-wise, we've got a virus there. About how big would my prokaryote be? Bigger. Yeah, so a virus is just generally like a little piece of RNA or DNA depending on the virus. So viruses are hard to see through a microscope. Prokaryotes or bacteria, you can see through a microscope. You can physically see them. Okay, so prokaryotic cell. And then that would be the relative size of the eukaryotic cell, more or less. So at least 10 times as big for a mammalian cell as for a bacterial cell. So if you're ever looking through, say in 4LL, you're looking through a microscope, you see tiny, tiny little specks that are swimming around in a non-random fashion. That's a bad sign if you're trying to grow your mammalian cells and you see those in your medium, because that means you've got contamination. So random specks maybe occasionally, but if they're like swimming consistently, that's not a good sign if you're trying to grow mammalian cells, because it means bacterial cells are in there. Bacterial cells, like an order of magnitude bigger. So they're really quite easy to distinguish under a microscope. As I said, E. coli, they're really the powerhouse of protein production in the bioengineering world. They're sturdy because of that cell wall, so they hold up to shear stresses. There's another thing that might not be immediately apparent, but not all cells, but many cells need, viruses not so much, viruses need other cells usually to live, but many cells need oxygen or even just other nutrients to live. So thinking about that, what's the difference between a prokaryote and a eukaryote? that? What's the consequence of that? So here you have a lot more surface area for a little bit of volume. This one you have a smaller amount of surface area to a bigger amount of volume. So what does that mean in terms of nutrient diffusion? Exactly, exactly. So it's much easier for nutrients, including oxygen, to diffuse what they need into the part of the cell that needs it. For prokaryotes, just because they're smaller. So eukaryotes, you have that whole diffusion barrier to get what it needs into the center of it. Okay. Yeah, so as I said, E. coli, really efficient little bugs, really easy to grow, not really good at making anything other than small, simple proteins without complex glycosylation. Okay. So if you need anything that you just need to make lots of, but it doesn't really matter that it's folded perfectly or glycosylated perfectly and it's not a really big protein, definitely go here first. So for example, one of the proteins that we had, you had the last year's class expressing in 4LL3 was expressed in E. coli, but it was kind of at the limit of what E. coli could express properly and fold properly, because their protein folding machinery is not as efficient as eukaryote animal cell protein folding machinery. And what the consequence of that is that while E. coli grows best at 37 degrees, when you express that particular protein, you had to express it at 20 degrees or lower. Otherwise, it would be trying to make and fold the protein too quickly, and it would not fold it correctly, so the protein would not be active. So it's sort of like a toddler who's trying to run and trips over its feet. The E. coli was tripping over its feet trying to make this protein, and it wouldn't be folded properly. So it can make slightly more, like once you start pushing the limits, you have to start really playing with what conditions it's making it under, how fast it's making it. As you can imagine, if you start trying to make it at 20 degrees, it's going to be making it much more slowly than if it can make it at 37 degrees. So you have a real trade-off in production efficiency by having a more complex protein. And then if you push it too much, it just can't make it, and you have to move to the much more expensive production system. Okay, so technically anything that you can see in a microscope is called a microbe, but it tends to include anything sort of single celled and below. So when people say microbe, they can even sometimes be referring to a virus, but typically not. Usually when we say microbe, we think of something that can exist or normally exists as in a single cell mechanism. So bacteria normally exist or can easily exist just in a single cell. But animal cells and plant cells, while we can culture them in a single cell suspension, don't normally exist in a single cell suspension. They exist as part of a more complex organism. You can argue that there are bacterial communities that are more complex, so when there are biofilms formed on surfaces, they form more complex communities. You have fungi that form more complex communities, arguably. They have form sensing and all of those things. But really, most bacteria normally are happy growing as single bacteria, not as part of a community and not as part of a larger organism, which is much more often seen for prokaryotes. Okay, so we said last class, 4 hours ago, that one of the big things that was the difference between producing penicillin penicillin at a low rate and a slightly higher rate was what it was eating. So the nutrients that it was getting. So what nutrients does a cell need to get in order to live? Thinking at the elemental level, what elements does a cell need?
Anything that is easy to make something, it's easy to get somehow, so carbon, oxygen, hydrogen. Some of the like chlorine, sodium, magnesium, those are used not as much though.
Sulfur, I heard someone say, what's an element that's used in DNA? Okay, sorry, I'll put magnesium up here. Also potassium. All right, these all count as macronutrients. They really only count as macronutrients because they are needed in large quantity by the cells. So those are the building blocks of the cells. Of course, carbon is the backbone. Carbon, oxygen, nitrogen, and hydrogen are needed to make proteins. Sulfur, what's the role of sulfur in a protein? Disulfide bond, cysteine, right? So a lot of the conformation of the protein, which we'll talk about in a second, is determined by sulfur. That's obviously needed in DNA. So all of these are sort of essential building blocks without which the cell can't survive, and they're needed in relatively large quantities. Also ATP, so your energy production is another big reason you need a lot of that phosphorus. So we've got also, I got told chlorine, that's an A, sorry, not a nitrogen. Sodium, what else? Calcium. Plus, minus. What else? That's a pretty good list. Okay, so I'll just add a couple of others. Zinc, molybdenum, and then, so these ones often, all the little metals, these ones and those ones, often act as enzyme cofactors. So an enzyme will often bind a metal ion sort of in the middle of it, and it keeps the conformation in a shape that the enzyme will work. Okay, so that's often why we need to use all of these metal ions, is because our enzymes won't work unless they have those metal cofactors in them. You also need, so if you think about what your mom says you need to eat, we're getting beyond, so these things often can be provided in close to elemental fashion. Typically you wouldn't eat coal, you'd eat some sort of carbohydrate as your carbon source, and that's true for prokaryotes as well. So some kind of carbohydrate tends to be, whether it's a simple sugar or something more complex, that tends to be where you get your carbon source. And along that, of course, with your oxygen and hydrogen, your mom always tells you, or your dad, that you need to eat your protein, especially get nagged if you're vegetarian. But what else do you need in your diet? What are the things that we can't produce in an elemental fashion? In addition to those metals and vitamins? Yeah, so vitamins. Can anyone think of anything else? Yeah, iron. That's another one. Yeah, so some amino acids. So I'm going to call that metabolic precursors, just because it's more all-encompassing. But absolutely, there are some amino acids we can't make. Carnivores, there are a number of amino acids they can't make, and that's why they need to eat meat, is because they are not capable of synthesizing some of those amino acids themselves, and they need to get them through their diet. Also, there are some growth hormones that cells won't easily metabolize for themselves. So these are all things that are important to think about when you're providing nutrients and a broth for your cells to thrive in. If they don't have some of those factors, some of those components in their medium, in their nutrient medium, then they won't be able to thrive. You won't have a good yield. They won't efficiently be able to make their product. So I actually had, when I was in 4th year, I took a plant physiology class, and the coolest part of that is that we had a lab that was in the greenhouse, and we were sort of personally responsible for 4 different plants, and they were grown hydroponically, and the first one had a complete nutrient solution for it to grow in hydroponically, and in each of the other 3, some nutrient was missing. So you had to, give it its fresh nutrient solution that was maybe missing something every week to make sure that it was happily growing hydroponically. And then you noted what happened to the plants when one particular nutrient was missing. You try not to do that to people. But people have discovered what happens when you are missing like specific things in diet. So like if you're missing vitamin C, what happens? You get scurvy. So there was a classic tale of a first year who only ate Kraft dinner for his entire first year and got scurvy. It was more typically sailors who didn't have fresh fruit and vegetables that would get scurvy, but that's why they started taking limes and lemons on board ships so they didn't get scurvy. And there are a few others that are caused just by single nutrient deficiencies, not ones that you typically see in the developed world, but that you very often see in sort of poorer circumstances, or just for first years who don't know what they're doing. So there are also, the other thing, of course, is that there are most cells need a carbon source, and so if you need a carbon source, that's called heterotrophic. So we're heterotrophic, most bacteria are heterotrophic, most cells in general are heterotrophic, but some, if they have, where'd it go? Chloroplasts are called autotrophic. So, need carbon. Okay. So they can fix their own carbon. Plants, algae also can fix their own carbon from carbon dioxide in the air. Okay, so it's not emerging from nowhere. They're not, plants aren't producing elements from nothing, but CO2 rather than a nutrient broth. Okay, and of course, oxygen typically from air. But autotrophs, although it seems more efficient, the whole process is much more complex. So typically their growth is much slower. If you're talking about growing cells in a single cell suspension, it's more difficult to do that in a single cell suspension with plant cells and algae, partly because they need sunlight, but they also need to diffuse CO2 in, and the whole process of fixing the carbon dioxide is fairly energy intensive and takes time for it to happen and work efficiently. So when you see plant cells being used, typically that's where people actually do root culture as opposed to trying to do a single cell suspension. And that's part of why it's just a little bit less efficient. But what we're trying to do is make a media that has most of those things in it, ideally all of those things in it, so that our cells will be the most efficient, have the highest yield of the product that we're trying to make. Okay, so we've got growth media. So that just means, media just means it's the broth that the cells are growing in. Okay, so there's some, if you've ever done cell culture, you'll see things called minimal media or minimal essential media, MEM. These are defined medias or media that have very specific carbohydrates and elemental things in them. They are put in, they are repeatable, and typically that's what regulatory agencies like to see, because they know every time that what has gone into the media that supports the growth of your cells is exactly the same as the last time you said, so that you're not doing anything unexpected in your process that you're not wanting to see. However, the minimal essential media, there's so pure, and specific amounts. You can imagine that's actually quite expensive to make and is probably not the thing that's going to give you the highest growth. Because if in fact you're having to include amino acids, you're having to include vitamins, you're having to synthesize growth hormones, those are really expensive to make in pure forms that last and last. Like that'll be good to put in your medium. They have shelf lives that are quite short. They're hard to purify and expensive to purify. So it's expensive to have a minimal or defined medium, and it tends to give you lower yields. So it's very rare in a lab setting for people to use minimal essential or minimal or defined media because it's not the fastest way to make your cells happy. So more typically, you'll see rich or complex media. And they might, so in animal cell culture, which we do a lot in my lab, you'll start with the minimal essential medium, and then you'll add stuff to it to make it into a complex medium that will happily support cell growth. So in animal cell culture, the usual thing that you put in is fetal bovine serum, or FBS. So typically in animal cell culture, you'll add 10% fetal bovine serum, sometimes 5, sometimes 20, depending on your cells and how picky they are. And what that has in it is lots and lots of growth hormones, lots of vitamins, lots of metabolic precursors, all the things that make it simple and easy for your cells to live. In other cases, you'll see yeast extract, and what's the problem with that from a regulatory point of view? So the cells are very happy with this. Why would the regulatory agencies not be so keen?
It's going to occur in individual cells.
Yep, so say your cows are fed one way in one part of the country and they're fed another way in the other part of the country. Your FBS will be different. Or, you know, they're exposed to pollution in China, or they're grazing on a field that has a lot of heavy metals somewhere else. Your FBS could be quite different in different locations. So there's variability that's introduced by having a natural component into your medium. So that's potentially problematic. And the same thing is true for yeast extract as well. The way you extract it, the way you've grown the yeast, all of these things can feed into your yeast extract being slightly different than the other person's batch somewhere else in the country. Or a batch that was produced in the high humidity of summer versus the winter time. Okay, so that's problematic. Why would regulatory agencies be particularly twitchy about fetal bovine serum? Yeah. So mad cow disease is transmitted by prions, which don't break down when you heat your medium up. So you can sterilize it all you want, but it will still have prions in it, which can cause mad cow disease. So FBS is actually very, very difficult to, mammalian cells are very, very difficult to grow without FBS. But there's been an increasing push by the regulatory agencies to try not to have it because they're concerned about transmitting mad cow disease through FBS. Interestingly, there's also a company that tried to, I think they managed, they were trying to start up last year. And in Canada, it's not legal to pay for blood donations because the idea is that if people are giving blood donations only for the monetary reward, they're probably reluctant to disclose if there are any reasons they shouldn't give blood donations. So if they have had hepatitis or they have other communicable diseases, they may still have the incentive to give blood and not disclose the problems if you pay money for it. But this company actually wanted to pay people to donate blood in Canada because what they were doing was using just basically human serum to make pharmaceutical products, avoiding the problems with bovine spongiform encephalopathy. And they were saying they're just using it to basically make pharmaceutical products, so it shouldn't have that same level of oversight on it. And I'm not sure, I need to follow up, actually, if you're interested, you can follow up and tell me next class, if they managed to get the approvals to do it. Their biggest argument was that when human serum is used in pharmaceutical company production in Canada, there's very little of it available from Canadian sources. Mostly it comes from the United States, where you are allowed already to pay for blood products. And so you're already being exposed to risks, it's just not from Canada. So that was their argument. Anyway, so it's much easier to grow things in rich media. There are also some that are called reduced media. The purpose of that is there are anaerobes that don't grow properly if there's oxygen around, so you add a reducing agent like cysteine, ascorbate, thioglycolate that removes the oxygen from the medium so that the anaerobes can grow properly. There are also differential and selective media. So has anyone ever heard of a differential medium or a selective medium, or can you guess what they might do? I'll see if anyone at CC has an answer. Yeah.
We came across one called XGAL.
So XGAL is typically a marker for that gets expressed.
You've used McConkey agar before?
Sorry?
McConkey agar?
McConkey agar?
Yeah.
So what does it do?
Gram positive or something?
So it would be a differential medium that would allow gram-positive cells to grow but not gram-negative cells. Yeah, that would be a really good example of a differential medium. So, oh, is it colors
Yeah,
one?
and then it also
Okay.
doesn't allow them to grow things. differentiate
So-
between gram-positive and negative, but it also selects for a certain metabolic complex.
Okay. Yeah, so that would still be a good example of something. So differential media will preferentially let one cell type grow, or color one cell type and not another. So it somehow differentiates between different cell types. So that would be a great example of that. How about a selective medium? So you were close with the XGAL suggestion. You were totally on the right track. So XGAL itself it basically just turns cells blue if they've been genetically modified with the XGAL protein, and you put it in the right stain that allows that to turn blue. But you're totally on the right track. Any other thoughts? Yeah.
With selective media, they allow a certain type to grow?
Exactly. So a selective medium typically will poison everything that's not the specific cell type that you want to grow. So typically that's used if you've genetically engineered an organism that confers like for example, most commonly antibiotic resistance. So you make your E. coli antibiotic resistant. If it has the gene of interest, then it'll be antibiotic resistant. So you put antibiotics in your growth medium that select for the genetically transformed cells, and everything else dies off except the ones that have been transformed to be genetically antibiotic resistant. So it's a really nice way to make sure that your cells stay as a pure culture, because bacterial cells in particular are really good at mutating and kicking out genes that you've tried to put in them. So we'll talk about this again and again in class. One thing that you have to be really careful about is if you put in genetically modify an organism to make lots and lots of your protein of interest, it now doesn't have the same resources to put into growing and multiplying. So if a small number of those cells kick out the gene of interest, they will overgrow your genetically modified cells because they can put all their energy and all their resources into growing and multiplying instead of making your protein of interest. So there it's really important to have a selective medium that will kill off anything that's kicked out your gene of interest. Okay, so those are the basic growth media. I'm going to go, how many people, the next part, I think I can do it in 6 minutes if you guys don't mind. I'm just going to go very quickly through the building blocks of a cell. I think you're probably all comfortable with it. I just want to review it super quickly so that everybody's on the same page and thinking the same way. But as you can imagine, you need all of those nutrients in order to build a cell. So amino acids, what are amino acids? You guys can tell me this. What's an amino acid?
Building block of protein.
Yeah, so proteins are made up of, they're linear polymers, and the mers are amino acids. So amino acids can have a lot of different properties that determine how the linear protein folds. So the littlest, anyone remember what the littlest amino acid is? Glycine. It just has a hydrogen. So glycine's kind of cool because that's where a glycine will happen at a lot of bends, because it'll be really flexible because it doesn't have any side chains to prevent the main polymer chain from bending around. So you'll see a lot of glycine at bends. There's some, typically they'll have different charges. So you'll have positive ones and negative ones. So you get ionic interactions between the positive and negative amino acids. What other properties can amino acids have?
It has aromatic rings.
Yeah, so if it has aromatic rings, does it tend to be hydrophilic or hydrophobic? So the aromatic rings can actually do two things. One is they're really bulky. So they'll actually cause a kink in the backbone, typically. So it'll make the backbone bend where there's an aromatic ring. But then we also have that hydrophobic, hydrophilic question. So mostly they're hydrophobic. So if you think about them, they're kind of like hydrocarbons, which are hydrophobic. So benzene would be an example of an aromatic ring, and it's, the way you can remember is that you get it in gas, and it's a hydrocarbon that's hydrophobic. It separates out from water. So a lot of the aromatic ringed ones are also hydrophobic, which means that you tend to find them on the interior of proteins and not on the exterior where they come in contact with water. And then the other sort of relatively special one that we touched on before has sulfurs in it, cysteine. So it'll form disulfide bonds that make a protein sort of stay in conformation, because disulfide, so most of what controls a protein's conformation are van der Waals forces, electrostatic interactions, and hydrophobic, hydrophilic interactions. But when you have, so they're non-covalent bonds, but the cysteines can form, if you have two cysteines together, they can form covalent disulfide bonds that really stabilize a protein structure. So cysteines can be important. So if you take out a cysteine from a protein, it'll suddenly, it can suddenly start folding very, very differently than it would otherwise. So there's some that aren't going to make much difference if you substitute a different amino acid in, and some that are going to make a really big difference. Okay, so we have what's called the primary structure. And that's the sequence of amino acids determines the primary structure of the protein. So every protein has a different sequence of amino acids that make up the protein, and that's called the primary structure. So does anyone remember what the secondary structure is? So I heard alpha helices and beta sheets again. So you've got these terrible electron alpha helices. Single-stranded helices. So the primary, you can actually predict oftentimes some secondary structures from a primary structure. So there are some sequences of amino acids that lend themselves easily to alpha helices, because what you're happy to see is there's whatever the side chains are of the amino acids. They tend to interact with each other from loop to loop of the helix, and that stabilizes the alpha helix. So you can, to some extent, predict just from this primary structure what secondary structures you have. Beta sheets too, to some extent, what you have is these interactions, electrostatic and other interactions between the beta sheets. You can have them actually like this, or you can have them do something else and then move around and then come back all in the same direction. That's the other way that beta sheets can form. So they don't necessarily have to be next to one another in the primary sequence to form a beta sheet, but they interact with each other. You also often see glycines on these hairpin terms because it's easier to bend there. So that's the secondary structure. Oh, I'm out of time, but I'm just going to finish up super quick. The tertiary structure and the quaternary structure. Anyone tell me super quick what those are?
Tertiary structure is function interactions, and quaternary is protein-protein interactions.
Yeah. So this is where you get subunits. So like in your blood, you get the 4 units that come together to hold your oxygen together. The quaternary structure is sort of the big blob shape that makes up the protein overall. So how it folds into a big shape, but that some of these will interact with other subunits of the protein and make up the quaternary structure. So are important in a bioprocess, because what you want to get ultimately is a functional protein at the end where your quaternary structure is appropriate with the highest amount of